In this part, I want to talk about the adaptive immunity. Recall that adaptive immunity is provided by the adaptive immunity system and involves T lymphocyte and B lymphocyte exclusively. The lymphocytes formed and the products they secrete in our body's defense are collectively referred as the immune response. The adaptive immunity response is initiated upon entry of foreign substances or antigen. However, it takes longer to effectively develop than the response which we have for innate immunity in our body. Lymphocytes must take contact with the antigen, which cause a lymphocyte to do proliferation and differentiate to form a specialized clone or army of the lymphocyte against that specific antigen. At least several days are generally required to develop an immune response upon the first exposure to the antigen. And because of that, we call the adaptive immunity the third line of defense. We have two branches for adaptive immunity. One of them is called cell-mediated immunity, which involves the T lymphocyte, and the other one is called humoral immunity or antibody-mediated immunity, and it involves the B lymphocyte, plasma cells, and the antibodies in the body. In this picture, you can see the adaptive immunity. As you see here, we have one cell-mediated immunity, which are T lymphocytes are active. They are effective against antigen within the cells, and they require antigen-presenting cell. One cell should get the antigen, engulf it, break it down, represent it to immune system, and then the T cells become active. We have two types of T cells. One of them is called T helper. First, it can detect antigen presenting cell, and then it hires different types of lymphocyte. Sometimes it can hire the cytotoxic T cells, and the cytotoxic T cells can fight with the antigen cell to cell, and as you see here, they can destroy the antigen by apoptosis. And sometimes they can hire the B lymphocyte. If they hire the B lymphocyte, the B lymphocyte cannot work directly on the antigen. They should first change to the plasma cell, and when they change to plasma cell, they can produce antibody. You need to know that the B lymphocytes are effective against antigens which are located out of the cell. It doesn't need the antigen presenting cells sometimes or most of the times, and the antibody which is secreted from them can work and fight against antigens. But what is antigen? The pathogenic organism and other foreign substances are detected by T lymphocyte and B lymphocyte because they contain antigens. An antigen is a substance that binds to a component of adaptive immune system, T lymphocyte or the antibody. Antigens are unique to each infectious agent and are usually proteins or large polysaccharide molecule. Example of antigen include parts of infectious agent such as the protein capsid of the virus, cell wall of the bacteria or fungi, and bacterial toxin. The tumoric cells also contain antigen. In the case of cancerous cells, mutation occurs that generally result in the production of the unique proteins, which we call them tumor antigens. We have different types of antigens. Some of the antigens are called foreign antigens. Foreign antigens differ from human body molecule, and we have some self-antigen, which are the antigen on the cell surface inside the body. So the foreign antigen or non-self antigen binds with the body's immune component because they are different enough in structure from the human body's molecule. In contrast, the body's molecule typically don't bind with the body's immune component, which we call them self antigens. Lymphocytes are generally very effective in distinguishing a self-antigen from the foreign antigen. And one of the malfunctions which we have in adaptive immune system is autoimmune disorders, and it happens when the immune system starts to fight with self-antigen. And we have some antigenic determinants, which we call them epitopes. But what are they? The lymphocytes normally have contact with only a portion of the antigen. The specific site of the antigen molecule that is recognized by lymphocyte is referred to as the antigenic determinant or epitope. Each type of antigenic determinant have a different shape, and a pathogenic organism can have numerous different antigenic determinants. An antigen that induce the immune response is more specifically called the immunogen, and its ability to cause an immune response is termed the immunogenicity. So immunogenicity is ability to trigger a response, and it increases with antigens degrees of foreignness, size, complexity, and quantity. And haptens are small foreign molecules that induce the immune response. When attached to a carrier molecule in host, like the toxin which we have in poison ivy is called hapten. It's different from the epitope. The hapten itself doesn't have antigenic activity, but when it attached to the carrier molecule in the host, it becomes antigen, and it can trigger and stimulate the immune response. And the autoimmune disease occur when the immune system doesn't have tolerance for a specific self-antigen, and we have immune response against that self-antigen. It initiates immune response as if the cells were foreign, like rheumatoid heart disease, diabetes type 1, and multiple sclerosis or MS, and all affecting different body structures. We have two types of lymphocyte in our body. We have T lymphocytes and B lymphocyte, and they differ from other immune cells because each lymphocyte has a unique receptor complex, which are composed of several different and separate proteins. There are typically about 100,000 receptors complexes per each cell. A receptor complex will bind one specific antigen. The antigen receptor of a T lymphocyte is called TRC, as you see in this picture, and the antigen receptor for B lymphocyte is called BRC. TRC is the abbreviation of T cell receptor, and BRC is the B cell receptor. And these receptors are the receptors for the antigen. The initial contact made between a TRC or BRC of a lymphocyte and the antigen, which recognize it. T lymphocyte must first have the antigen processed and presented in the plasma membrane of another type of cell. T lymphocytes simply are not able to recognize the antigen without this step. In contrast, B lymphocyte can make direct contact with an antigen, and it doesn't need any cells as the host of that antigen. We have two different types of T lymphocyte. T lymphocytes have additional receptor molecule that facilitate T lymphocyte physical interaction with a cell-presenting antigen. One significant category of co-receptors is the CD molecule. In fact, the two major types of T lymphocytes are T helpers and T cytotoxic, can be distinguished based on the CD protein which they have on their surface. The T helper lymphocyte contains CD4, and they assist in cell-mediated, humoral, and innate immunity. They can activate natural killer cell and macrophages, and cytotoxic T lymphocytes contain CD8 on the surface of their cell. They can release chemicals which can destroy the other cells and push these cells to do apoptosis. Other types include memory T cells and regulatory T cells with their specific surface proteins. What are the antigen presenting cells, and what is the meaning of MHC molecule? The antigen presentation is the display of antigen on a cell's plasma membrane surface. This is a necessary process performed by other cells so that T lymphocyte can recognize the antigen. Generally, two categories of cell present antigen to T lymphocyte. All nucleated cells of the body and a category of cells which we call them antigen presenting cells. The term antigen presenting cells, or APC, refer to immune cells that present to both T helpers and T cytotoxic cells, and they include dendritic cell, macrophages, and B lymphocyte. Antigen presentation requires the physical attachment of antigen to a specific transmembrane protein. This transmembrane protein is called major histocompatibility complex, or MHC. The MHC is a group of transmembrane protein, and we have two types of MHC. MHC1 is found on the surface of all nucleated cells, and MHC2 is found on the APCs, in addition to MHC1. And antigen presentation requires the physical attachment of antigen and this MHC on the surface of our cells. In this picture, you can see the formation of lymphocytes. You know that in the red bone marrow, we have a lymphoid stem cell, and this lymphoid stem cell can differentiate to lymphoblast. If the lymphoblast remain in the bone marrow and mature in the bone marrow, they can make B lymphocyte. If these lymphoblasts migrate to thymus, they change to T cells and they mature to T cells. And we have two types of T cells. One of them is called T helper, and the other one is called T cytotoxic. And you see that the secondary lymphatic structures can house the B lymphocyte, T lymphocyte, both of them, the T helper and T cytotoxic. And these are the sites which these lymphocytes become activated and do proliferation. Here you can see the B cells which change to plasma cell, and the plasma cell can release antibody. And here you can see the T cells or T lymphocytes. First we have T helper, and this T helper become activated and it can hire the cytotoxic T lymphocyte. And this cytotoxic T lymphocyte can release some chemicals and it can do apoptosis of the pathogen for us. The lymphocytes are formed in the red bone marrow, as I told you, and they are then tested for immunocompetence. Immunocompetent cells bind and respond to antigen. So first, maybe our T cells or B cells are not active. They cannot respond to any pathogens. But when they become immunocompetent, it means that now they are active and they can bind and respond to a specific type of antigen. And this testing occurs during the development and shortly after birth. And it occurs in the primary lymphatic structures. And we know that the primary lymphatic structures are the lymph node, are the thymus and the bone marrow. The B lymphocyte can make the humoral or antibody-mediated immune response. They have the specific receptors, and by these specific receptors, they can bind to the specific antigen. This antigen doesn't need to be presented by antigen-presenting cells. The binding event activates the lymphocyte to undergo some clonal selection or doing proliferation. And a large number of clones are produced, and this is one of the humoral response which we have in our body. And let me talk about this slide on this picture. Here in this picture, you see that the antigen come to the body, and this antigen can attach to the surface protein which we have, or surface receptor which we have on the surface of our B cells, which we call them B cells receptor or BCR. Not all of our B cells have that receptor for that specific antigen. So as you see here, we have some receptors with green color, some receptors with blue color, and the antigen cannot attach to them. But for this dark blue color, the antigen and the surface receptor can bind together. When this binding and attachment take place, we have proliferation of the B lymphoblast, and these B lymphoblast can make a large number, and they can make the accumulation, which we call it the clone. Most of the cells in the clone can change to plasma cell, as you see here. The plasma cell is a cell which is differentiated from the B lymphocyte, and it has this potential to secrete antibody. And some of these B lymphocytes remain as the memory cell in our body. They don't change to plasma cell. And in that second exposure to that specific pathogen, the memory B cells remember the first exposure. So they can immediately make the clone, they can immediately respond to this antigen, and they can change to plasma cell, make more antibody. And in the second exposure, you see that our response can be very stronger, and also we can make more antibody than the previous and the first exposure. And what about antibodies? What are antibodies? The antibody is an immunoglobulin or Ig protein produced against a particular antigen. The structure of antibody is very similar to letter Y, and it has two parts which can attach to the antigen, which we call them antigen binding site. You can see the shape of the antibody here. It's like letter Y, and in these two parts we have a site which can connect to antigen. We have different classes for antibodies. One of them is called IgG, and the IgG is predominant antibody in the lymph, in the cerebrospinal fluid, in the serous fluid and peritoneal fluid, and it's about 75 to 85% of the antibodies in our body. The other antibody is called IgA. IgA can be found in the areas which are exposed to the environment, such as mucous membrane, tonsils, and they can make different secretion. I mean the mucous membrane can make different secretion like mucus, saliva, tears, and breast milk. And IgA plays a significant role in protecting the respiratory system and gastrointestinal tract and also urogenital system. The other classes of antibody is IgM, and we can find IgM in the blood. The other one is IgD. Again, we can see it in the blood, and also the IgD is the antibody which can easily move from mother to fetus at the site of placenta. And the last one is called IgE. IgE is formed at the time of allergic reaction, and IgE causes release of histamine and other mediators which can make inflammation for us. So these are the different classes of antibody which we have in our body. But what about the cellular or cell-mediated immune response? The antigens must be presented by macrophages to the immunocompetent T cells. I told you that the T cells cannot work directly with the antigen. First, one cell, like macrophage, like dendritic cell, should catch the antigen and break it down and represent it to immune system and T cells. T cells must recognize between self-antigen and non-self antigen. So their function are very important. And after antigen binding, they can make colon. The colon is very similar to the colon of B cells, but different classes of cells are produced here. Here you can see the cell-mediated response. You see that first we have macrophage, gets the antigen as you see here, and it represents the antigen on its surface, and then the T helper become active. The T helper become active, can identify that type of the antigen presenting cell and the antigen which is present on the surface of this cell, and then it can hire T cytotoxic cell, and T cytotoxic can attack that infected cells. And sometimes our humoral immunity become active and the B cells change to plasma cells and start making antibodies. And we have, as I told you, we have two types of T cells, two important types of T cells. T cytotoxic, they are specialized in killing infected cells. They insert a toxic chemicals, and these toxic chemicals is called perforin. If you look at this picture, you can see this cytotoxic T cell, and this is a cancerous cell. These T cells can make perforin, and perforin can make these pores on the surface of the cells. And after that, the water and salt and fluid can come, and the cell become bigger and bigger, it's swollen, and then it bursts. This is how the cytotoxic T cells can work in our body. And the T helper cell is the cell which can recreate or hire the other cells to fight with the invaders. And they can interact directly with our B cells too. And besides the T helper and the T cytotoxic, we have the other T cells which we call it suppressor T cells. They release chemicals to suppress the activity of B cells and T cells. And it happens when all the antigens in our body are destroyed, and they stop the immune response, because if the immune response is not stopped and if they have uncontrolled activity, they can remove and destroy the other cells in our body too. So their functions are very important. And now you can answer these two questions. What is the HOP10? What is the difference between HOP10 and the epitope? You need to know these differences. And where does a lymphocyte usually become activated? And how many types of lymphocyte do we have? And in each type, how many subtypes can you find? The other part which is important to know is immunologic memory and immunity. And you know that for that we need to do vaccination. sometimes, and sometimes we can acquire immunity naturally. So the acquired immunity which we have in our body is divided to two types of immunity. One of them is called naturally acquired, and one of them is called artificially acquired. The naturally acquired immunity is divided to active and passive immunity. It means that in naturally acquired, our body is active to make its own antibody. Sometimes when we exposed to a specific pathogen, or it may be passive when the antibody pass from the placenta, from mother to fetus, and come to the body of fetus and bring immunity for that. But sometimes we have artificially acquired immunity. In artificially acquired immunity, sometimes the immunity is active when we do vaccination, and sometimes it's passive. It means that we use the specific type of antibody from the other person directly to the body. So we have two types of acquired immunity, naturally acquired and artificially acquired immunity. And in these slides, I talked about these types of immunity, which I talk about it in this slide again too. The active and passive immunity are different. First, I want to talk about the active immunity. Active immunity happen when we can produce the memory cells due to the contact with the antigen. Sometimes this active immunity is naturally acquired when we are exposed to antigen, and sometimes it's artificially acquired when we can get the antigen by vaccination. And the passive immunity, we don't make the memory cells in our body. In naturally acquired, the antibody can passively move from body of mother to the body of fetus, or we can find it in the breast milk. And the artificially acquired passive immunity happen when we use antibody from the human or the animal to the person, and we can immune it from different diseases. And vaccination is the weakened or dead microorganism or component. It can stimulate our immune system to develop memory B lymphocytes, and it can provide relatively safe means for initial exposure to microorganism. And later we exposed to that microorganism, the secondary response which is created is much more stronger. The immune system response predominantly from the humeral branch, and the B cells become active here, and they can provide lifetime immunity, like for example chickenpox vaccination. But sometimes we need to do a booster shot, like the rabies, like tetanus, like influenza. We need to do the booster. And we have a disease which we call it acquired immunodeficiency syndrome, or AIDS. This problem is a life-threatening condition that is the result of the human immunodeficiency virus, or HIV virus. And HIV infection targets the immune system, in particular the T helper cells, the cells which contain CD4 on their surface. HIV infects and destroys these T helper cells over a period of time, and prolonged HIV infection leads to devastating effect of the AIDS. And we have different means for transmission, like intercourse, needle sharing, and breastfeeding, and prevention happen by the safe sex. And HIV test looking for HIV antibodies in the blood becomes AIDS when T helper cells drop their number in our blood in a certain level, and individuals prey to opportunistic infections and may have many CNS complications. We don't have any specific treatment for HIV, and only we should prevent the spreading of this virus. And in this part, you can answer these two questions. How do vaccinations related to immunologic memory, and what are the different sources of passive immunity? You need to remember that we have two types of immunity. One of them is active, when our body actively make antibody. Sometimes we can acquire it naturally, and sometimes it's artificially. And the other type of immunity is called the passive immunity. And the passive immunity, sometimes naturally we can get the antibody from our mother, or we can get it from the breast milk, and sometimes it's artificially acquired when we use the antibody of the animals. You need to know that in active immunity we make memory cells, but in passive immunity we don't make memory cells.